Hi, I am Nicole Murdoch, and today I'm going to be talking about CRISPR-A or CRISPR activation. So first, I want to go over how CRISPR-Cas9 works and how it has been used previously for gene editing before I go into the modifications that have been made in order to make CRISPR activation work. So the only two parts that are really necessary in any CRISPR system is a Cas9 protein and a guide RNA, which the guide RNA then composes of a tracer RNA and a CRISPR RNA. So the tracer RNA is a section of RNA that base pairs to the section of DNA that we want to focus on or change. And then the CRISPR RNA is a palindromic sequence that binds to the Cas9 protein. And then the Cas9 protein is able to cut the DNA where it's lined up. So the steps that happens, the guide RNA recognizes it, it lines up the Cas9 protein, it cuts the DNA, and then scientists can go in and modify that DNA by either changing out bases if there's like a mutation, or they can delete things or create insertions. Also, one typical way of how they insert this into organisms or cells is through plasmids. So they have one plasmid for the Cas9 protein and also one other plasmid for the RNA. So how do we modify this for CRISPR activation? Well, first, the purpose of activation is going to be to activate specific genes to transcribe them. So in order to do this, we don't want to cut the DNA anymore. So you have to go in and do 2-point mutations in the Cas9 protein plasmid in order to deactivate its endonuclease subunit. And luckily, this does not change its ability to bind to the DNA or guide RNA because those subunits are still functional. And then through creating a fusion protein, including the Cas9 protein, activation domains are added to recruit transcriptional factors. These transcriptional factors can be, well, there can be a lot of them depending on which activation domain we're using, but typically it'll be your TF2D complex and almost always, well, your RNA polymerase. So one example here, it's a pretty simple one. One of the first they came up with is the VP64, which you can see the VP64 is attached to the dCas9 protein. And the RNA polymerase is then there transcribing the gene. Now you can add a lot of different activation domains. Here is an image of a few different ones that have been used in different research to test their functionality, and they've all worked. They just work slightly differently for different genes. And there's a lot more than this. Just give you a little overview of different possibilities. So just to look a little closely at two options out there, there was first the VP64, in which there was a study where scientists thought that it might be more activated if you were to add more proteins on top of it. So then they created the dCas9 VPR, in which there's also a P65 and an RTA protein attached to the Cas9. And they ran some tests on this, and studies actually showed that adding the extra proteins greatly increased the transcription activity. Also, I was going to little side mention, as you can see here, they are actually doing like a lot of studies right now on just optimizing the system and trying to figure out how to best use CRISPR activation as opposed to using it too much in research, but I will get a little bit into some studies that have used to learn more about genes later on. But here is another study done by Horizon Discovery, in which they wanted to see if adding multiple CRISPR RNAs would increase transcription, and it in fact did. So the way that they did it, they tested on EGFR and POU5F1, and they tested with 4 different CRISPR RNAs and then a pool of all 4 CRISPR RNAs for both of them. And well, for the first example, you can see that it greatly increased the transcription having the pool. And in the second one, it didn't increase it in every case because it matched the CRISPR RNA one, but it wasn't detrimental to it by any means, and it was greater than all of the other CRISPR RNAs. So, how can we actually use this technology? How is it going to be beneficial or going to help us learn more things? Well, first off, if there is a gene that we want to learn more about and see what its function is, if we activate it, we can run some tests, see if there's phenotypic changes or changes in concentrations of anything within the organism that will help us learn a lot more about what that gene is. Also, if we want to do that in the reverse process, if there is a phenotype or a mutation that we want to go in and fix, but we need to figure out what the gene that codes for it is, we can activate a few different genes that we may think may possibly code for that and figure out where the mutation is coming from. And then one other way that has been used is you can use it to determine like upstream and downstream effects to see how one gene regulates other genes. So one study that was done on the upstream and downstream regulation done by Horizon Discovery was that they took IL-1R2 and specifically activated just that gene to see how it affected other genes, and they expected that it would inhibit the transcription of both IL-6 and IL-8, but it would, in contrast, activate the transcription of ZEB2 and GEMIN2, which, as you can see here, it did in fact do that. So the transcription of IL-1R2 was much higher than it would have been if the activation was not there. And then the IL-1/6 and IL-1/8 have much lower transcription. And then the ZEB2 and the GEMIN2 ended up with higher transcription. And then one other interesting study that I found that they have been using CRISPR activation tools to make their study work a little better is this one in which they're trying to use Streptomyces species to find new antibiotics, because as we get more antibiotic resistance out there, we need to come up with new antibiotics to continue to fight off bacteria. And they suspected that in BGCs, biosynthetic gene clusters, there would be more antibiotics that we have not found or do not currently use. But the only issue with trying to do more research and transcribe them to learn more about them is that they are not easily expressed in the lab because they are only expressed under certain conditions. So they used CRISPR activation to activate those genes, and we're actually successfully able to create 2 CRISPR-Cas9 systems to be able to activate silent BGCs and learn more about the antibiotics they could have. And here is a list of the sources that I use. Thank you.